It's a true honor for me to introduce Roderick Eckenhoff. I think everybody in anesthesia knows him because he is one of the world experts on the mechanisms of action of anesthesia, of anesthetics. So he doesn't need an introduction. So why am I here? Because there is a special link to him. I'm working at Northwestern, and those of you who are who have lots of experience, I was almost going to say are older. I'm not going to do that. Go on, where are you? So those of you with certain experience know the book Trips, Eckenhoff, Van Dam. That was a standard textbook of anesthesia. The Eckenhoff, the author of that book, was Roderick's dad. Now what's so special about it is that Eckenhoff was the first chair of anesthesia at Northwestern University. People still talk about him because he was an effective leader and he was a gentle person. He quickly became dean, and I must say everybody loved him. And again, they still talk about him. So Roderick is one of the very few persons who has also become a giant in anesthesia, succeeding his father. It doesn't happen very often. And the other thing I want to mention is that he also inherited the gentle personality of his dad. So, Roderick, thank you so much for coming.
Thank you so much, Jan, Andre, and the other members of the NAVAt team for inviting me here. I must admit I've never been to Belgium before, at least in my memory. And although my dad had to travel through here once, and he may have towed us along, so I can't remember. But in any case, it's a real pleasure to be here. Thank you. So the topic today, anesthetic mechanisms via target discovery. We heard a little bit from Chris earlier and yesterday about the different levels of anesthetic mechanisms. You can look at it at the imaging level to see what the brain is doing. You can look at it at the electrophysiologic level and EEG and see what the brain's doing electrically. You can look at it at the neuron level, as Chris is doing, and see what's happening at a little deeper level. But today we're going to dive even deeper. We're going to go to the molecular level because that's the level that we really need to understand if we're going to make our drugs better. And so that's what I've been focusing on for the last, I don't know, 25 years, 30 years or so. So disclosures are these, funded mostly through NIGMS and NIA. This is an interesting organization which is trying to develop new volatile anesthetics, which is interesting. I haven't heard from them in about a year, so I don't know how they're doing. And these are the molecules that we're going to be talking about today, and a few others. So ranges from xenon to sevoflurane and everything in between, the historical ones like halothane and cyclopropane and so forth. So the real question that has been the focus of this field for the last 50 years really is, where are these drugs acting? What are the molecular targets for these very small drugs that seem to share no structural similarity? They share physicochemical similarity, but not structural similarity. So where are they acting? Are they acting at the same site? Is there a unitary mechanism for anesthetics? And the answer to this question is it depends. It depends on how we look for these mechanisms and for these targets. And so these are some of the methods that have been used for target discovery for the drugs that you all use. I'm assuming most of you are anesthesiologists, that you all use in your practice every day. And so I'll go through these one by one, but in vitro sensitivity, which I call guess and check, in vivo sensitivity, in vivo regulation, and specific binding. So to go through these in a little more depth, in vitro sensitivity. So the assumption underlying this approach is that the activity of these targets will be influenced in a plausible direction at a concentration similar to that we use to anesthetize our patients. Okay? So people will choose their favorite target. This is what I mean by guess and check, and then see if it reacts, if it changes its activity in a plausible manner. So The targets satisfying this assumption are many. This is going to be a continual theme here this morning. Chloride, calcium, potassium, sodium, proton channels, water channels, all kinds of channels respond to anesthetics in an appropriate concentration. Receptors, the GPCRs, ligand-gated ion channels, those are channels 2, N receptors, enzymes, ATPase, cyclase, etc., second messengers, even the structural proteins in our cells, and I'll talk a little bit about some of the motor proteins, which I think are particularly interesting. What about in vivo? So that was in vitro sensitivity, where you express a channel in your oocyte and you study it with patch clamping and so forth. What about in vivo sensitivity? So the assumption here is that manipulation in either the amount or the activity of a given target or pathway will influence the anesthetic sensitivity, whatever endpoint you want to use. It will influence the anesthetic sensitivity in an organism, in an intact organism. Okay? So what are the approaches here? They can be pharmacologic, as shown here. Gaba-A, satisfying the pharmacologic approach. Opioid, alpha-2, dopamine, cholinergic, histaminergic, KB, and etc. So there are many, 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 many channels satisfying this assumption as well. Genetic. What if we alter genetically an organism and see what the effect is on anesthetic sensitivity? What has satisfied that criteria? Mitochondrial complex 1, syntaxin, GABA-A channels, 10-pore potassium channels. Again, there are many. This is going to be, again, a continuing theme throughout this, and I hope that's something that sticks, is that there's no unitary mechanism for the drugs that we use to put patients to sleep. This is what I call the origin of the 20% rule, is that most of these manipulations change anesthetic sensitivity by about 20%, maybe 30% in most cases. In the most extreme case, and that's the 10-pore potassium channels, by about 40 to 50%. Never complete, and they're not additive. So you can't make two manipulations and change it in by 60% or 70% or that kind of things. Okay? I call that the 20% rule. In vivo regulation. The assumption here is that the organism will regulate either the expression or transcription of those targets most affected by the anesthetic in an attempt to restore homeostasis. This is the most tenuous of these approaches, but it's something that we've explored a little bit way back when, and that can be genomic or epigenomic or proteomic approaches. And just quickly to go through some of these results. So this is in rats exposed to either halothane 10 exposures. Every other day we give them a 2-hour exposure to halothane, or fewer exposures, 5 exposures. We compared that to isoflurane. And then we looked at the transcripts, the mRNA. This was something that was very popular a few years ago. And you looked at the transcripts that were altered in the brain by this manipulation. And first, we found that isoflurane didn't change anything. If you do the proper false discovery rate statistics on these many different transcripts, you find there's no effect whatsoever. Probably because this is the Achilles heel of this approach. You need to have an enormous effect to satisfy the false discovery rate criteria. Halothane changed a few, but just a few. And they weren't very interesting. And so the transcription results from this in vivo regulation approach, few genes, little overlap with the known, in quotes here, because known is mostly from guess and check approaches, targets. And the possibility is that these are the important targets that I just showed you. That's really unlikely. Activity is not altered enough to invoke regulation. To me, that's the most likely. Or regulation occurs post-transcriptionally at the protein level. So let's look at that. We've looked at that as well. So the approach here is a little different. Instead of doing the mRNA transcript analysis, you do proteomics. You grind up the brain, run it out on a 2-dimensional gel, and then you can compare in the same gel. You can label them differently and compare them in the same gel. And this is a quantitative proteomics via differential in gel electrophoresis called DIGE. It was very popular for a while. It's not so popular now. And when we did that, we looked at different regions of the brain, different anesthetics. We could detect over 2,000 different proteins with this approach. And the ones that were matched with the control gels, over 1,000. And we ended up with a fair number of spots that were changed. Okay, so this indicates, well, there is some translational regulation that's going on here. And the summary of these results is here. 5 to 10% of what we could detect were changed. Mostly after the anesthetic, not during. If we did it immediately at the end of the anesthetic, we didn't see any change. If we looked 2 hours later, then we did see some changes. Halothane produced largely changes in metabolic proteins. Isoflurane, stress-related proteins. There was little overlap between the anesthetics. We'll come back to this because this is a very interesting thing. Most of us think that halothane, isoflurane, sevoflurane, it's eeny, meeny, miny, mo, right? You get in there in the OR in the morning and you just choose which vaporizer you're going to use, and frankly, it doesn't make a lot of difference. They are different. That's a message I hope that you'll take away from this, is that they are different. They have different fingerprints of changes in the brain and even at the molecular level. Again, no overlap with the known sensitivity targets. And we also did isolated cells, and they showed mostly structural and stress-related proteins. So again, not a lot of clues here as to what's going on, except for the fact that perhaps there are a lot of different targets involved. So all of the regulated targets, again, regulated by the organism, targets, they could be indirect effects. We don't know if the anesthetic is affecting that target directly, or we don't know if it's an upstream effect that's producing changes downstream. So they may not be drug targets, and how do we find out if they're drug targets? We test for specific binding, and this was the last approach in the different approaches for detecting anesthetic mechanisms that I mentioned, right? How do we do that? Enter photoaffinity labeling. So this is something that we started back almost 30 years ago now. And we started with halothane. And why halothane? Halothane has the very unique feature of having a carbon-bromine bond. None of our other anesthetics have that. And why is that special? Because it's UV labile. In other words, if you shine UV light of a specific wavelength on halothane, it'll cleave apart at the carbon-bromine bond, leaving a free bromine ion and a carbon-centered radical. Okay, so what? Why is that interesting? Because this carbon-centered radical will then bite into whatever it happens to be near and covalently adduct to it, become part of it. Why is that important? Because if we put a carbon-14 label on this, we can now tell what targets it's actually attaching to. So we did that. So this is work done by Mary Ellen, sitting right there. So the first thing we did is ask the question, where in the brain are anesthetics affecting? Are they affecting everywhere? Is it a global phenomenon, or are there some unique features, like, for example, the sleep center in the hypothalamus? Are there some specific, or the thalamus, specific regions? So this is an autoradiogram of a rat brain exposed to carbon-14 halothane that's labeled with photoaffinity labeling. Okay? Everything that you see here that's dark is where it's bound. This panel is if you add in about 10-fold higher cold halothane, unlabeled halothane. Why did we do that? Because that's how you test for specific binding, whether or not this binding is pharmacologically specific. And interestingly, most of it is. So the first answer you get out of this is most of that there's no preferred regions, although there were some interesting regions in the cerebellum, but really no preferred regions, and most of it was specific. That almost sounds like a contradiction, but you'll find out that it's not. Interestingly, and again, getting back to the regulation results that I showed you a minute ago, if you add in excess cold isoflurane, there is no competition. So that argues that most of this labeling, most of this widespread labeling is actually different than where isoflurane binds. So these drugs truly have different targets and a different flavor of receptors. They could be the same receptor, but a different site within that receptor, and we'll show you an example of that in a minute. Chloroform, which is a similar haloalkane to halothane, this is a haloether, haloalkane, is intermediate. Okay, so it partially, and that makes some chemical sense. And if you take those brains that I just showed, you grind them up, and you run them out on these cells, you find out that there are a lot of binding targets according to this approach, about 15%. It's about the same number that we saw for the translation results, about 10-15% of targets of proteins that we could detect are in fact binding targets for this drug. Okay. Now, the photochemistry of halothane is messy, and it's messy for 3 reasons. First of all, the light required to activate that carbon-bromine bond is about 250 nanometers. 250 nanometers is short UV. It's the bad stuff. And it damages proteins, it damages the targets that you're trying to look at, so it makes the subsequent biochemistry that you're trying to detect really challenging. So that's number 1. Number 2 is this carbon-centered radical has a half-life of about a microsecond. And I know that because we actually measured it. About a microsecond. Now that sounds pretty short, but a microsecond, a molecule can diffuse a very long way in a microsecond. And so that makes it uncertain as to whether the target site, the site that you actually found it on, is actually where it was before you activated it. And so that's a problem. The next problem is that this bromine ion is itself reactive. And so these two can then react with each other. This can react with the target, and it can make the photochemistry very messy. So halothane was not a good photo label. So we went down the street. Nice thing about Penn is the unified campus. I can go literally across the street to the Department of Chemistry, find world-class chemists who do all kinds of crazy stuff with molecules. And what we did then is we made a series of molecules incorporating this group here. This is called a diazarine. And so we have examples of barbiturates, we have examples of propofol, which I'll talk more about in a minute, sevoflurane, isoflurane, desflurane, and so forth. And so we have examples, photo labels of all these molecules now. So I'm going to talk first about propofol, because this is where we've done a lot of work in the last several years. So the chemistry whiz that I have is this guy, Bill Daly. This is work done by a medical student, actually, Michael Hall, who just finished his anesthesia residency. And what we did is take propofol, we put this group onto it here. There are a couple other centers that have done this. This is one done in England by Nick Franks. This is one done up in, I'm sorry, this is Nick Franks, this is Keith Miller. But this is the one that we made. And the way this works with the diazarine is a little different than with halothane. So you take your protein of interest. It can either be an isolated protein or a complex mixture, a gamish. You equilibrate it with this molecule here. You hit it with light. Now, the big advantage of the diazarine is this, 370 nanometer light instead of 250. That does not damage proteins. This is long UV. This is the good UV, and so this doesn't damage the molecules that you're trying to understand afterwards. So that's a big advantage. You hit it with that. This now, instead of a carbon-centered radical, creates a carbene. A carbene is a very reactive molecule. It's 1,000-fold more reactive than a carbon-centered radical. So it reacts with its targets in nanoseconds. And so that is, it cannot diffuse very far, because if it doesn't have an attractive target that it can bind to, it's going to bind with water. So it's a very powerful approach to understand where these molecules work. And so then, depending on whether you put a radioactive tag on here or you use mass spectroscopy, which is what we've been using now for a while, you can then detect where this is, not only what proteins it's attached to, but where in the protein it's attached to. So, as I just mentioned, You can find sites within targets or targets themselves. So we're going to start out talking about purified targets. We're going to say, okay, we know from the literature, a vast literature, that the ligand-gated ion channels such as Gaba, glycine, and so forth are sort of canonical targets. We think that those are very important targets for some of these drugs, especially for Propofol. So we're going to start out talking about those, and we're going to start with GLIC. Why GLIC? What is GLIC? GLIC is a ligand-gated ion channel that's found in bacteria. It's homologous to GABA and glycine and the mammalian ligand-gated ion channels. And the big advantage is that it's been crystallized, so we know its structure. And further, it's been crystallized in the presence of propofol and desflurane. Okay, so that's very unusual. We have very few examples of membrane proteins that are crystallized with the drugs that we use every day. So this is what it looks like. It's a pentamer. Okay, it's a homopentamer, which means all the subunits are the same. And it forms an ion channel here in the middle. It's got this transmembrane region and this extracellular region. It conducts protons, largely, and anesthetics inhibit it. Okay? That's crucial to recognize. Anesthetics inhibit this protein. Okay? What do they do to GABA? They tend to potentiate GABA. We'll get to that in a minute. So they have opposite effects, and that's very important. So we found, to just go right to the conclusion here, that there was a good agreement between the crystal structure and the photolabeling. So here is propofol in the crystal structure, showing that the pocket that it binds to, these are the residues. These are the residues that we bound to using photolabeling. And so molecular modeling using these residues as anchoring points puts it in exactly the same place. So, okay, so this is kind of validation for our approach and for this particular molecule. And I should go back here to here and just point out that these blue blobs are where that site is. Okay? Now, if you look, this is the transmembrane region of one subunit. This is another subunit and another subunit. It's intra-subunit. I may get intra and inter mixed up, so just I'll try to get it right. But this is intra-subunit. It's in between the transmembrane sequences of a given ion channel, and that's important. Again, inhibited ion channel. And so good agreement there. We look then at the nicotinic acetylcholine receptor. Anesthetics do what to this? They inhibit it in general. And using the same drug, propofol, we found the same thing. But there are three classes. We had some complexity here now. This is no longer a homopentamer. This is a heteropentamer. There are two alphas, a beta and a gamma, delta, sorry. And the sites are, there's a pore site. Well, that makes perfect sense. It's inhibited. If you occupy the pore, that should inhibit it, right? But we also have these intra-subunit sites as well. Again, an inhibited ion channel. There is also evidence for an inter-subunit site. So a little bit of complexity when you start mixing up the subunits. Now we get to the one that everybody likes to talk about, and that's the GABA-A receptor. Again, this is another ligand-gated ion channel. It's a heteropentamer, but a lot of the studies that have been done so far are with homopentamers or with an artificial subunit composition. So, and we're going to start with that as well. So this is just an alpha-beta construct. And we find now, using again the same molecule, we find the same I'm sorry, I told you I'd get that wrong. We use the same inter, we see inter subunit sites, okay? Now, why is that important? Because this is a potentiated ion channel. So it would appear that we could begin to attach functional significance to where these sites are. If it's inter-subunit, we think that indicates that it's a inhibited ion channel. If it's inter, it's potentiated ion channel. Now, What I just showed you were receptors that we either express and purify in detergent or purify from a natural source, but it's in detergent, okay? So it's artificial. Does that reflect what's actually going on in a real neuronal membrane? How do we do that? These are very non-abundant ion channels. You can't do a gel, a typical SDS page gel of a brain and see a, and see a band where GABA should be. You won't see it because it's very non-abundant. And how can we see that? So we thought about this for a while, and we came up with this strategy. This is incorporating what's called a click moiety into our photo label. So now this is a bifunctional photo label. This is the click moiety, okay? Triple bonds do not exist in biology. And so by using this, first doing the photo labeling with this group, and then using this with this copper catalyzed click chemistry, we can attach all kinds of things to this triple bonded group. We can attach fluorophores, we can attach affinity tags, we can attach all kinds of stuff once this is covalently bound to the sites of action. So why is that important? Because that allows you to enrich for whatever it is that you've tagged. If you use avid and biotin chemistry, you can enrich your moiety, your group. Whatever, you take a whole brain, grind it up, you first do the photo labeling, and then you select, basically, using avid and biotin chemistry, you select the groups that you've labeled specifically and then look at them. So this is a very powerful way of enriching your targets. This is just to show that it works the same, has the same, you got to prove all these weird compounds that you make are in fact anesthetics. And so we do that with tadpoles typically, and we show that it has the same activity as Propofol. It's actually amazing that you can take Propofol and do just dozens and dozens and dozens of changes to the basic molecule, and it's still an anesthetic. It's really remarkable. Wouldn't give it to people, but it's still remarkable. So when we did this, and we looked specifically for GABA receptors in what we pulled out, we find that the alpha and beta subunits have a very large enrichment factor. This is an enrichment factor over what you put in as to what you get out. So this shows that these in fact are in vivo targets of the drug. So it sort of clarifies our results a bit from the purified receptors. These are in fact, and this just shows you that the degree of competition or specificity of the binding event here, that it's fairly high. And so, okay, they're actual targets in vivo as well as in vitro. And so this is kind of a summary of what I've showed you so far, is that the inhibited ion channels have transmembrane intra-subunit sites, maybe some core binding. The potentiated ion channels, such as GABA-A and probably glycine, although we haven't looked at glycine yet, has a inter-subunit site. Alpha-beta subunits are preferred. Now, why do I say that? All I pulled out was alpha and beta subunits. We looked specifically, and we looked very hard for gamma subunits. For Propofol, didn't find any. So that argues that the alpha gamma and the beta gamma interfaces in a natural synaptic GABA channel. GABA channels in the synaptic space are typified by their alpha beta gamma heteropentramers. Okay, we never saw anything binding to gamma, so that's actually interesting, and we'll come back to that in a minute. Okay, so now I'm going to turn away from Propofol and talk about, I think, what may be of more interest to this group, and that's the inhaled drugs. They're the most challenging. The chemistry, they're simple molecules, so it would seem that it would be pretty easy to make these photo labels, but it's incredibly difficult. And it's very difficult to then purify them once you make them. But Bill, again, was a wizard at this. And then this is an incredibly talented graduate student who's now a postdoc at the University of British Columbia. Keith Miller and Manuel Covarrubias did the electrophysiology. So these are the compounds that we've made. For isoflurane, this is AZ-iso. For SIBO, this is AZ-SIBO. We've already validated these as anesthetics. They work in tadpoles. They do things to GABA electrophysiology, shown here. just like their parent compounds do. They potentiate. AZ-CIVO, interestingly, is a little more potent than CIVO as an anesthetic. Again, I don't think I'm not recommending it for use. So they're just like their parent anesthetics. And where do they go in a native GABA receptor? So this is work that was just published in the FACEP journal. So all I can say, the easiest way to summarize this is it's complicated. There are lots of different sites. On average, for the inhaled anesthetics, there are between 8 and 10 sites on GABA, on one GABA receptor, 8 and 10 different sites. And the sites are mostly in TER subunit. Couple messages to relieve with you. Inter, that's one message, they're inter-subunit sites largely. Isoflurane and sevoflurane don't always occupy the same site. Now this gets back to some of the earlier slides I showed you that suggest that these actually are different flavors, that these molecules. We shouldn't be eeny, meeny, miny, mo when we select an anesthetic. There may be specificity of action and side effects to these drugs that we don't yet appreciate, but they're different, and they bind to different sites, or at least non-overlapping sites. There may still be an inter-subunit site, but it's at a different location at that inter-subunit site. So that's an important message that we should take away from this. Now, the other interesting thing about sevoflurane and isoflurane is that they do bind at the gamma interface. So this is unlike propofol. We didn't see any evidence for propofol binding at these gamma subunits, but we do see evidence for isoflurane and sevoflurane binding there, and at an intra-subunit location. Okay, so this gets complicated, right? Because I said that intra is inhibiting, inter is potentiating. So there's this battle going on between sites for the same molecule in a given receptor. Perhaps, as you all know, propofol has an ED50 of about a micromolar. It's very potent. Well, as anesthetics go, it's very potent. Isoflurane and halothane have an ED50 of about 200 micromolar. Perhaps this is why they're less potent. Perhaps because they bind at an inhibiting site in a potentiating, so there is this self-antagonism that's going on within the same receptor. And this is just one receptor. They bind to lots of other things, as you're going to see here. So other things that we captured from that click experiment that we talked about, other things that we captured were the potassium channels, sodium channels, the HCN channels, and a lot of others. So these were not potentiated to quite the same degree, but fairly good. And so these are targets as well. So we have looked at some of these, particularly the voltage-gated ion channels. So this is your classic KB 1.2 potassium channel, voltage-gated channel. They're very sensitive to anesthetics in vitro. A lot of work, Hugh Hemmings has done this, Mike Alkire, Emmanuel Covarrubias have done lots of work with these, showing that they are sensitive to anesthetics. They're plausible in the direction of change. In fact, if you put a KB 1.2, I think it's KB 1.2 antibody in certain areas of the brain, you can wake an animal up. So just an antibody, which is really kind of interesting. So they are plausible targets. So if you take the wild type, you do electrophysiology, most of them inhibit, so this is an interesting channel, most of them inhibit. Sevoflurane potentiates a little bit, okay? But then if you play with it, and this is work that Manuel has done over many, many years, if you play with a particular area, there's an area in this channel called the S45 linker. It links the voltage sensitive domain to the pore domain. It's a hinge region. Okay, so it's very, very important in linking the function of the voltage sensor to the pore. So if you play with that and you do some mutations there, you find out that it converts the inhibition to potentiation of most of the drugs. And for sevoflurane, it's huge. So this seems to be a very sensitive site. The question is it a specific binding site? This is done through mutagenesis. Is it a binding site for the anesthetics? And so, make a long story short, yes, It is. In the KV1.2 wild type, we find this leucine, which is a adduct site for propofol and for isoflurane. And depending on the mutation, this same site is, so let me just summarize this quickly. This site, independent of what it's doing functionally, is a binding site for the drug. What does that mean? So that means that when you do mutagenesis of these proteins, you are changing functionally the entire protein. You are changing the energetic barriers between different states. And so you can occupy the same site in this altered receptor and do a completely different thing. That's really important for those of you who are electrophysiologists and do mutagenesis, that you have to confirm that that site is a binding site with some direct technique like photolabeling. So just to summarize these labels, these are labels in the S4-5 linker. Interestingly, this is at the bottom of the transmembrane domain, not like it was in the Gaba channel. It doesn't matter whether it's what receptor, what potassium channel it is, you get binding there. Okay, so in the last few minutes, do I have a few more minutes? In the last few minutes, I'm going to talk about other targets. These are ones that you probably haven't thought about or haven't heard about. Okay? So, kinesin, tubulin, structure. These are structural proteins that we typically don't think about as anesthetic targets, right? SIRT2, LFA1, and many, many, many more. I'm only going to scratch the surface. We all know about the RYR, right? The ryanodine receptor. It's MH. It's sensitive to most of our inhaled anesthetics, but not propofol. That's a target, and we're trying to understand that at the moment. But this is one that I found particularly cool. So this is kinesin. This little guy here, we'll see it again here. This little guy here is kinesin. It's dragging a vesicle along a microtubule. So these are hugely important for axonal transport. These are hugely important for mitochondrial transport within an axon to the terminal. They are the trains that carry substrate all around the cell, all up and down axons, all around the cell. Hugely important for activity. And why am I showing you this? Because this woman published an abstract. This is Susan Gilbert. Published an abstract showing, and she just on a suggestion during this seminar that she tried with propofol, and it was enormously sensitive to propofol. And so we banded together and looked at this, and it turns out what's happening with propofol, it doesn't change the speed at which the train is going along the tracks. Has no effect on velocity, but they fall off. So they'll be chugging along and then just fall off. Whereas normally they'll chug along for a fairly long distance along a microtubule, but propofol significantly shortens about 30 or 40% at low micromolar and high nanomolar concentrations. So this is a very, very, very sensitive effect. And so we're trying to understand what's going on here. We think we've worked out the mechanism. The binding site is in this area of the foot. Let me just go back here. The feet are down here. These are what's called the motor domain. It's not going to play. But so down here in the foot is a binding site. And what happens is that as the foot goes forward and grabs onto the microtubule, the power stroke is a neck linker stroke. It comes back and it docks the neck of the foot to the body of the kinesin. And so that power stroke is really important. Propofol is right here. Propofol prevents that docking of the neck linker, and so it just falls off. It can't go anywhere. It gets into a sort of a struggle and falls off. So we think we understand the mechanism. This work was just published recently. Oh yeah, okay. Here is this movie showing. This is the neck that connects it to the attachment domain, and so these things. Every step is an ATP, and so this is this little neck linker part that's got to dock in as it does this power stroke across here. It's just so cool. And you can watch these with single molecule studies. You can have it drag a little nanoparticle along, so you can actually watch a single molecule of this stuff chugging along. And again, the effect of propofol and sevoflurane, we've done this now with sevoflurane, is the same. Sevoflurane's a little less potent, as you would expect. And so what could this subserve? Well, we've seen that long-range connectivity through all these wonderful imaging studies and the EEG studies and so forth. Long-range connectivity is lost. There doesn't seem to be crosstalk between areas of the brain that there used to be in the awake state. So perhaps these effects on the motor proteins could subserve some of that. This is work that we've got to do in the future, but that's just a hypothesis at this point. A lot of off-pathway targets, lots of off-pathway targets that we really don't know at this stage what they do, but I cannot believe they don't do something. SIR2 is a deacetylase, a very important regulatory protein enzyme in cells, and we're sure that it's doing something. We think it might be doing something during development because it's located in myelin and may have something to do with myelination defects as a result of anesthesia at a young age. Don't know the answer to that yet. LFA1 is one of these immunoproteins that allows attachment of monocytes and leukocytes to vascular membranes. It inhibits those as well, and so it may have effects on innate immunity, which we pretty much know it does. This is work done by Koichi Yuki at Boston Children's. This is a previous graduate student. And so, summary. Most general anesthetics are promiscuous. They interact with lots and lots of targets, and this is something I don't think people know, and that is that they interact with many sites within those targets, giving rise to enormous complexity. Targets for different general anesthetics may only partially overlap, no unitary target. All sites and targets are not functionally aligned, so you can have it interacting with, as I mentioned with Gaba, with different sites within a target that are not functionally aligned, and you can have different targets that are not functionally aligned. Stereoselectivity, cutoff, non-immobilizers, these are all things that you probably heard about in the literature over the years. They add only marginal relevance to this discussion, and I think they're mostly epiphenomena. Lipid membranes have still not been ruled out. For those of you who are lipid enthusiasts, lipid membranes are still there, and they're still important, and membrane proteins don't work so well without them. So we do tend to think that there are actions in the lipid membrane that could still be relevant and important to this. Summary 2, this is what I call small effects at multiple targets. I think the reason that we don't see a lot of regulation is because these are small effects. The organism doesn't know they're happening, and so it doesn't try to regulate them. And it's consistent, so the small effects of multiple targets is consistent with all of biology is sensitive to general anesthetics. We just heard about C. elegans, right? We've heard about plants, Venus flytrap, all those things are sensitive to general anesthetics. All of biology, down to bacteria, are sensitive to general anesthetics. Pressure is the only antagonist that I'm aware of that actually inhibits the effect of general anesthetics. And it also works in all of biology. Steep hill slopes. So what I mean by that, for those of you who do dose-response curves in populations, the steepness of that slope, defined by a hill curve, defines the variation within a population. And your typical narcotic would have a hill slope of 1. For anesthetics, it's 20, 30. There are no other drugs in our armamentarium that have that characteristic. None other. That means there's very little variation. I can pick any one or two of you out there and use the same concentration of anesthetic, and it'll work just fine. That's really uncommon for drugs. And so the reason that we think is because it has a small effects at multiple targets thing. Any given SNP or mutation or change in your genome, is not going to have a big enough effect to change your dose response curve because there are 100 other targets that are contributing. Adaptation doesn't occur. You can turn on isoflurane, and maybe many of you do this. You can turn on isoflurane for a 12-hour case. You can do it in the ICU for 2 weeks. They do this in kids to tolerate ECMO because the volume of other anesthetics they have to give gets progressively larger. And they don't change. You don't have to touch the vaporizer dial. That's also uncommon in biology. And a large variety of very simple and very dissimilar compounds can produce the same state. And so to me, this is what's going on. Discard the unitary hypothesis and start thinking about complexity, because really that's what this is. And this is Ray Fink. Anybody, you remember Ray Fink from way back? Yeah. He was a genius because this is how he characterized anesthesia. There's an underlying hidden multiplicity, and I think that's what I've just told you. So this is a group that I've been working within for a long time. This is our program project group. Manuel, Jan Shu at Pitt, Mike Klein, Grace Brannigan, who's now at Rutgers, she might be moving soon, and Kent Blasey, who's now at Irvine. Thank you.